So we're going to talk about the pancreas for a moment. So the pancreas is going to produce what we call pancreatic juice. This is a combination of both liquid and enzymes. So we just call it juice. And it's released into the duodenum. So that first part of the small intestine. Remember, small intestine goes duodenum, ileum, jejunum, ileum. So it's about a liter a day. So what is composed? It's the enzymes. Exocrine secretion is going to be pancreatic secretions because remember it's an exocrine and an endocrine gland. So these are going to be digestive enzymes is what we're talking about. So digestive enzymes and buffers. Mainly the buffer is going to be bicarbonate. So water is going to be a major component as well, ions, electrolytes, and then majorly it's going to be those things. So part of the pancreas, we talked about the islets, which are going to produce the endocrine portion, so the insulin and the glucagon, and those types of hormones. Now we're talking about the other part. So what is releasing into ducts instead of just the bloodstream? So it goes into ducts and then it goes into the major pancreatic duct and into the duodenum. So acidine cells are going to be the ones kind of surrounding the duct producing the enzymes. They produce what we call proenzymes because just like when we saw pepsinogen has to be activated as the inactive form of pepsin, we also have inactive forms of these enzymes. Why? Well, we don't want them to start digesting until they get the duodenum and get to the food that they're supposed to be digesting. You don't want all of a sudden them to start digesting proteins and carbohydrates and things that are inside the pancreas, because then you're going to be digesting your pancreas, and that's not a good thing. So we turned amylase again, starch. We had salivary amylase, which starts the process of digestion of starch in your mouth. And now we have pancreatic amylase, same thing, different production. So this is produced by the pancreas. Proteolytic enzymes. Proteolytic means protein digestion, so break down protein. These again are going to be released as inactive proenzymes because you want them to be inactive because you don't want them digesting the proteins of the pancreas. And then they're activated in the duodenum once they reach the small intestine, that first part, and they are meant to digest. So examples of those guys are trypsin, chymotrypsin, carboxypeptidase, and elastase. So all of the N's and A's over here, and then trypsin and chymotrypsin. You also have pancreatic lipase. We're going to talk a lot about that when we get to lipid digestion, so breakdown of fat. We also have nucleases, so nucleic acids are going to be broken down by these guys, break down RNA and DNA that's found inside cells. And then this is a major component. So all of these are the enzymes that are released by the pancreas. But another major component is sodium bicarbonate. Why is sodium bicarbonate released? Because it's a buffer. And so when sodium bicarbonate needs to kind of neutralize the acidity. So it's going to make it more neutral, so closer to 7. The chyme. So remember we call this chyme is coming out of the stomach into the duodenum. Well, the stomach is meant and capable of handling pHs of 1 to 3 with its hydrochloric acid. The duodenum, not so much. So you need sodium bicarbonate released by the pancreas to increase the pH. We want to increase the pH of what's coming from the stomach. If you do not do that, then you can cause erosion of the inner mucosa of the duodenum and cause ulcers. So without sodium bicarbonate or not functioning, then you're going to get duodenal ulcers and damage to the duodenum because of that damaging pH. So you want to actually increase the pH using sodium bicarbonate. Small intestine. Small intestine is all about surface area. So you're going to have different ways of getting surface area. So you have what we call circular folds. So these are things that you can see. So you'll see in lab that there's kind of these folds within the small intestine that you can see that are called circular that go all the way around. They are permanent, they're always going to be there, and they help swirl. Why is that important? Mixing. So it makes a bumpy road. If you think you have circular folds, you make a bumpy road. So something's going over it, well, then it's going to mix more because it's going to kind of move with it. And you're going to help kind of make it so it's not a smooth ride, it's a bumpy ride. So you want to mix. Why? Because you're adding all your digestive enzymes and that sodium bicarbonate from the pancreas. So especially in the duodenum, you want mixing because you want to mix all those enzymes and that bicarbonate with the chyme that's coming out of the stomach. Also, microscopically on the kind of surface of the intestine, you have villi, which are going to be large kind of projections that you're going to see that are going to come out. These are going to be where absorption's occurring, and you have more surface area because you can absorb more here, kind of going around all of these hills, versus if you just had a straight line. So this is where you can absorb things, and that's the point of the small intestine. Finish digestion and then absorb. So break things down and bring it into your bloodstream or your lymphatic system. Microvilli we've seen on cells, so they are microscopic too. That you can see on the surface of the cell. We also call it the brush border, so you're going to see brush border enzymes that are actually there. This is going to be ultimately where microscopic absorption happens, so things are going to go into the cell. Yeah. So things are going to be able to go into the cell that way. There. So enterocytes are these cells. So enterocytes are going to be the epithelial cells with the microvilli, the brush border. You also have crypts. So down in the valleys is going to be intestinal crypts. You are also going to release more water. This is why we have to absorb it back. And mucus, so you actually see goblet cells in this area that is going to help. So remember, we made a paste with the stomach, and we're adding more liquid to it so that we can help move it along. So goblet cells making the mucus. You always have stem cells because enterocytes are always going to be damaged and need to be replaced. So you'll have stem cells here, and it's the conveyor belt that you saw sort of in the epithelium of the skin. So the epidermis, it's going to go and then travel up as cells are dying and then replace them. So 1 to 2 liters of intestinal juice, again, it's giving me a lot of water that we're going to reabsorb in the large intestine, helps moisten the chyme. This includes more buffering, so not only the sodium bicarb from the pancreas, but the small intestine adds some more buffers to calm down the acid of the stomach as well. And then this is also helping with mixing. So again, chyme is more of a paste. We make it more into the liquidy so that we can have enzymes and everything mixed together well. If it's a paste, it's harder to mix the enzymes in. If it's a liquid, you can swirl it around with those circular folds and have mixing so the enzymes are getting exposed to everything that they're supposed to be breaking down. So remember brush border enzymes, like I said, so brush border is the microvilli on the surface of the cell. And we're going to see coming up later that the last part of breaking down things before they're absorbed actually happens at the microvilli where we have enzymes. So we have maltase, sucrase, lactase, nucleotidases, peptidases and dipeptidases. So maltase breaks down maltose, sucrase brings down sucrose, lactase breaks down lactose, nucleotidases is going to continue to break down RNA and DNA, and peptidases, dipeptidases is going to finish off proteins. And again, we'll look at those more when we get to upcoming videos. So the small intestine also has a couple of reflexes. And so kind of look at the breakdown of the word. Gastro means stomach. And then enteric is going to be small intestine. So once food is going to hit the stomach, so that you know food's going in, then the small intestine is going to get ready for it. So it's just a preparation. So you feel food extension in the stomach, then your small intestine is going to start moving things out. And if it's continuing, so it's going to stimulate motility along the entirety of the small intestine, and it's going to start secretion. So that's going to be intestinal juice. All of those things that we talked about just now is going to be starting to be produced and released. Your small intestine is going to start moving, segmentation and peristalsis is going to help occur because it's preparing. We know food's a coming, so we need to clear it out, make sure anything that's still in there gets out of it so we have room, and we know it's coming, so it's preparation time. Gastroileal, so the ileum is the last part of the small intestine. This is still stomach, so this is going stomach to ileum. This is making sure we're getting stuff out. So if we need more material to move from the small intestine to the large intestine, because that's where it happens, ileum leads to the ascending colon. So we have a valve in between it. It's called the ileocecal valve. So this is going to be, the ileocecal valve is going to be an area or sphincter kind of looking thing where we can open it and material can go from small intestine to large intestine. This is clearing things out. If you have food in your stomach, well, more's coming down. So you need to clear out space and make sure it goes into the large intestine so that you are ready for food to come farther down and be digested. Large intestine, after all the nutrient absorption and the digestion happens, then you receive the things that can't be digested, still chyme, not absorbed, not digested. The large intestine has a couple of things. So it has what we call haustra, which are kind of little pockets. So you can see, if you see picture, it kind of looks a little bit like that. It's got pockets combined to it. Each pocket is called a haustra. And there's a line of smooth muscle that's going to be in the muscularis layer of the large intestine that's going to be kind of going the length of it. It's called the tinea coli that scrunches and makes the haustra. That's going to come into play when we talk about movement of the large intestine. Another major component of the large intestine is there's a lot of bacteria. Lots and lots. There's over 1,000 plus different types and different species of bacteria that inhabit the large intestine. And they actually have you have more bacterial cells than you have human cells in your anus or your large intestine. So I was reading anus. So 10 to 1 of bacteria cells to a human cell. They either come up or down, so they either go from the anus and colonize the colon from the kind of anus to inside, or the small intestine come down being eaten. A lot of times the bacteria are going to ferment, so they can actually do a different type of digestion. So fermentation is digestion in a different way than humans can. So carbs and different things that we cannot digest, your small intestine can't digest, can occur by the bacteria that are in your large intestine. So they can actually, a lot of them, we think bacteria bad and we must get rid of them. There's beneficial bacteria. So these are going to be helping with breaking down carbs like plant material a lot of times that we can't digest. And fermentation does release some gases and some acids. So some sort of flatulence can occur because of this fermentation. So depending on what you eat and how indigestible for us it is, but digestible for the bacteria can produce a little more gas and flatulence. Another major component of the bacteria is it's going to make B complex and vitamin K. So things like B12, and there's B6 and other things. But we talked about B12 helping with cell division, needs intrinsic factor that's made by the stomach to absorb. So having this made by our bacteria is helping with cell division as well. So we are really looking into gut bacteria and how it can affect um, the health of somebody. So we actually show that sometimes gut bacteria can help with weight, overweightness, obesity, and things like that. That's why we can take probiotics, prebiotics, and things like that. Can help prevent some diseases like diabetes, atherosclerosis, and fatty liver, and even mood can be affected by gut bacteria. So they're really, really important. So we call kind of chyme that's undigested and not broken down by the bacteria called residue, and it hangs out in the large intestine for about 12 to 24 hours. Nothing with our large intestine, so your large intestine is not breaking down food. Nothing's happened anymore. Unless it's the bacteria. So your human cells can't break down anything, but bacteria can digest, again, those things that your cells can't digest, the undigestible for us. So those B vitamins and vitamin K that are made by the bacteria can be absorbed. Water is going to be a major place where you're taking that water back in that you've added from the pancreatic juice and the intestinal juice. Electrolytes, anything that you've lost or added, are going to be absorbed again. So sodium and chloride are going to be major ones that you've added. Again, intestinal juice, pancreatic juice, and you take them back. We don't want to lose those. We need to use them again. So the major function of the large intestine is to move feces to the anus for defecation, so to help remove waste. Also, remember bilirubin? So that's going to be the pigment from hemoglobin. So breakdown of blood cells, and you break it down into bilirubin. It breaks down bilirubin into what we call urobilins and stercobilin, and that's what's going to help. Again, it's pigment kind of variation, but that's what makes feces brown. Their brown color normally is the breakdown of bilirubin into urobilins and stercobilins. So what happens in the large intestine moving and moving feces? So we have those haustral contractions. So that is going to be contractions of those pouches that are happening. So this is slow moving because you have little contractions and they respond to stretch. So stretch out because there's material in here. So then they're going to contract and move a little bit to the next. And then you have stretch here, they're going to contract a little bit and you move to the next. So it's going to be slow movements, not much. It's going to take a while. You need something extra. And this is happening mostly in the ascending, meaning going up, and the transverse going to the side. The major thing that's going to happen, the major movement of feces is going to be secondary to the gastrocolic reflex. So it's another reflex. Food in stomach is going to stimulate the colon. So food in the stomach is going to cause what we call a mass movement. meaning all of a sudden you're going to have really big wave of peristalsis. So remember, that's our forward moving movement. So you're going to have, these are kind of small contractions, you're going to have big contractions. And these are going to happen 3 to 4 times a day after eating, depending on how much you're eating. And it's called the mass movement. So again, you have the ascending transverse and descending sigmoid. So all of a sudden, this is going to be a really big push forward to get feces to move. And that happens again after eating, especially 3 to 4 times a day. The descending colon and sigmoid colon are more storage reservoir. They're just waiting, kind of opportune time, have a mass movement that moves it forward enough into the rectum, and then we'll talk about the defecation reflex later on. Or not. So again, mass movement is going to push feces towards the rectum. So what happens again, distension, these are going to be pressure receptors, is going to stimulate a reflex in your spine. So it's going to be central nervous system. This reflex is parasympathetic, makes sense. Remember, this is rest and digest. So this is going to cause the last part of the colon. Remember, you have ascending, transverse, descending, sigmoid, and then the rectum. So you're going to stimulate contraction of the sigmoid to go into the rectum. You're also going to relax the internal anal sphincter. That's going to be smooth muscle. And that's involuntary. So the skeletal muscle and voluntary is going to be the external anal sphincter. So you're actually going to have the urge to defecate, but you won't go until you're ready. As long as everything's working okay, so you can consciously go to the restroom and relax your external anal sphincter and defecate. So you can see sensory nerve fibers, afferent nerve fibers go to the spinal cord. And then because you're feeling stretch receptors, so feces are moving into the sigmoid colon, stimulate stretch receptors. That is going to show reflex to the spinal cord and come down. And you're going to have the relaxation of the internal anal sphincters, and that's going to have feces come down. And then once you're ready, you do have input to your cerebral cortex to say, hey, you do have to defecate. And then you have a motor input to the external anal sphincter because you need both relaxed. So this is already relaxed and you're just waiting for the external to relax. So when it's convenient to defecate, then you voluntarily defecate because this is going to be part of conscious control or voluntary.